Welcome to the course Biochemistry 1 conducted by myself, Dr. S. Dasgupta of the Department of Chemistry, IIT Kharagpur. In this course, we will be studying certain aspects of biochemistry starting from the structures and functions of biomolecules right on to bioenergetics and metabolism. The topics that we will be covering are structures and functions of biological molecules. In that, we will be considering amino acids and proteins, enzymes. Under enzymes, we will be considering the mechanisms of specific enzymes. Then we will go on to vitamins and coenzymes, carbohydrates and lipids, nucleic acids and their components. All of the topics of this course will be covered in their entirety as is relevant to this course. We then will be considering the principles of bioenergetics with special reference to carbohydrate metabolism. The books that we will be covering are common biochemistry books such as Stryer, Leninger, Voigt and Voigt. Apart from this, there are many internet sites and websites that give us a lot of information on biology and biochemistry in general. So, you could also look up those sites where there are specific course material available and specific tutorials and quiz materials also available for you to access. When we consider the central dogma of biology, the first thing that comes to mind is DNA. DNA is the storage medium. The central dogma of biology goes like this: DNA to RNA to protein. We have a text that is comprised of DNA, which is the 4 bases of DNA that are the storage medium. This is then transcripted to RNA, which is the transmission medium, that also is comprised of 4 bases, one of them being a bit different. The RNA is then translated to the protein. The alphabet of DNA, RNA and protein is slightly different. What is this alphabet? In DNA, we have 4 letters to the alphabet. The 4 letters are, as you can see here, A, G, T and C. These are the 4 letters that comprise the alphabet of DNA. If you look at the corresponding alphabet of RNA, you will see that we have U, C, A and G. When we go on to study the structure and the contents of nucleic acids, the structures of each of these bases will be much clearer. But for now, we have to know that DNA and RNA are comprised of these letters, which are actually represent nitrogenous bases. The protein alphabet is a bit different. The protein alphabet is sometimes represented as a 3-letter code, which we will see in a moment, or by the 1-letter code, which is another representation of the very same 3-letter code. The protein alphabet is comprised of 20 unique letters that tell us what the protein sequence is. We will understand what an amino acid sequence is once we get into the details of what is the peptide bond and what is an amino acid. The first thing that we know or we try to understand here is the carbon atom. Now, if we consider what these proteins are actually made of, the proteins consist of amino acids that are linked by peptide bonds. We will understand this also in detail to see how a peptide bond is actually formed and how we can link these letters together into forming what would be like a sentence. Each amino acid consists of a central carbon atom, which you can see marked as C alpha here. So, we have the central carbon atom that is marked as C alpha. To it, we have an amino group. All of you recognize an amino group as the NH2 group. We then have the carboxylic acid group, which is COOH. We also have what is called an R group. Now, this